So following along with our central dogma, we go from DNA to RNA to protein. So in previous sections in Chapter 11, we have replicated DNA. We have already talked about transcription, going from DNA to RNA, and then this last piece is translation. So here in 11.4, we're going to be talking about translation. So remember, translation is a protein. So it's protein synthesis from mRNA transcripts. So remember that translation, just from previous courses from eukaryotic translation, we are utilizing our mRNA transcript that we just made. We're utilizing our ribosome, which includes our rRNA, and then we are utilizing our tRNAs to bring our amino acids to our growing chain of amino acids. So this includes our table of individual codons that are going to tell us which amino acid is going to be brought to the subunit. So recall that we have two subunits in our ribosome. As a part of our ribosome, we have the large subunit and then we have the small subunit. And then in here we also have our rRNA. So this is our large subunit, obviously, and small subunit. And then we have our mRNA here, mRNA transcript. And then we have our tRNA. Our tRNA has our 3-letter anticodon, letter meaning nucleotide, and then it looks like a T, actually, which includes those nucleotides. So this is on the end here is where we're going to have our amino acid attached. And then we have our anticodon, which are 3 nucleotides that are going to match up with 3 nucleotides on our mRNA called the codon. And this is all happening within the ribosome. So if we remember the specifics of it, we have our initiation. And initiation is where we have our complex forming. So all of these parts that we just talked about here are all going to be coming together. So our tRNA, this one here, our tRNA, is going to be bringing our first amino acid to this polypeptide chain, and then it's going to bind to the start codon on mRNA, and that's going to be within one of the spaces of our large ribosomal subunit. So if we kind of draw this out a little bit more specifically, we have our mRNA transcript here, and then within our large subunit we kind of have these sections, or these positions. And so these positions are going to allow for the tRNA to be in here and attaching to the different codons. So our start codon is always AUG. So if we look at our specific codon here, we would be coding for AUG on our mRNA. So those are 3 nucleotides, AUG, which means that our anticodon that's going to be coming in and matching up is going to be here, UAC. So UAC is going to be part of our, we're just going to kind of go up here. This is obviously part of it, part of our tRNA. And then on the end of this here, we have our first amino acid. Our first amino acid is going to be methionine. So, actually, I'll make this green here. First amino acid is methionine. This is our tRNA with our anticodon matching up with our codon AUG, which is always our start codon. So then what happens is we're going to shift our places here. So this ribosome will go this direction, and or we can say the mRNA is going to be moving in this direction with the tRNA still attached to it. So then we move this tRNA into this position here, which then allows a new tRNA to move into this middle position here with its new amino acid coming along. It's going to make a peptide bond between the methionine and the second amino acid. So if we can picture, this has moved over. This is our UAC because this one is now something else, UCC or something, because our AUG was over here, and then now this is AGG. So this is our methionine, this is some other amino acid, and now we've made a peptide bond between the two. Then this tRNA is going to go off, and we have a bond between these two that's going to keep growing. So we're going to keep adding more and more amino acids as we move along, shifting our mRNA through and moving our tRNAs and releasing our tRNAs. This all happens until we get to, so that's elongation. This is going to continue happening until we get to termination. And the way that termination works is that we have something called a release factor. And our release factor basically is just going to kind of jam the system here. So it recognizes the stop codon. So remember, we have a stop codon. We actually have 3 stop codons, UAA, UAG, and UGA. Those are all 3 stop codons. So once we reach the stop codon, we have what's called a release factor is going to actually attach to the stop codon as kind of an anticodon. It's not an anticodon, but sort of like an anticodon. And it's going to attach in there, and then it's going to cause the whole thing to dissociate. So the ribosome is going to fall apart, the mRNA is going to be released, then the peptide chain is going to be released as well. And so that ends our translation. Now, this is something that you are likely familiar with because this is the process that you've studied with eukaryotic organisms. And we see the same thing in prokaryotic organisms with some differences. And so we're going to talk about those differences now. So in our eukaryotic organisms, we have our ribosomes. And our ribosomes in eukaryotic organisms are 80S in the cytoplasm. And 70S in the mitochondria and chloroplasts. Now, these are not the ones that we are focusing on so much, but these 80S ones that are in the cytoplasm are the ones that our mRNAs are mostly going to be going to. So that is different from our prokaryotic organisms, where their ribosomes are always the 70S. So this is going to be a huge thing to keep in mind moving forward, so make sure that you know this information, although it seems small right now perhaps, when we get to things like how antibiotics work. Again, this is a very big difference when we talk about antibiotics, because we can have antibiotics that are going to attack the 70S ribosomes, and what that's going to do is it's going to cause problems with our prokaryotic organisms, but not our eukaryotic organisms, because we have the 80S ribosomes. Another difference is that our initiator in eukaryotic organisms, our initiator tRNA is going to carry methionine. And then in prokaryotic organisms, our initiator tRNA is going to carry N-formal methionine. So very similar to methionine, but slightly different. And again, these differences matter when we're trying to get rid of pathogens and not kill our own cells. So the initiator tRNA carries N-formal methionine and not just plain regular methionine. Another difference is that in our eukaryotic organisms, the ribosomes are going to bind to that 5' cap. So it's looking out for the 5' cap, and then it's going to track to the start codon. And then in our prokaryotic organisms, the ribosome is not going to bind to the 5' cap because there is no 5'. Excuse me, there is no 5' cap in the mRNA transcript. Remember, we don't have RNA processing. So it's going to bind to what's called the Shine-Dalgarno sequence. So this is also called ribosomal binding site, obviously named for the people that studied it. And it is an AGGAGG. And then it's going to track to the start codon. So very similar in that we have the 5' cap here that's the ribosome will bind to, because it says, okay, once it finds this 5' cap, it says, okay, this is mRNA. We're going to start to come together and we're going to do this whole translation thing. Versus prokaryotic organisms, it finds this Shine-Dalgarno sequence and says, okay, this is mRNA, we're going to come together, we're going to do this translation thing. And then it'll track to the start codon in either case. But again, since we don't have RNA processing in prokaryotic organisms, we don't go to the 5' cap, we go to the Shine-Dalgarno sequence. So those are three differences between eukaryotic and prokaryotic organisms in translation. One thing that also needs to be mentioned is that in prokaryotic organisms, we see both transcription and translation taking place at the same time. So I'm going to put a picture in here that is from the textbook on that. All right, so we see that picture here. So in the upper left-hand corner here, we can see this is our prokaryotic organism. And you can see the cell here, the plasma membrane with the cytoplasm. We have transcription occurring here, so because this is the DNA. We have transcription occurring here with the mRNA being made. And then we have translation happening at the same time. So right here, these are our ribosomes. They have them labeled here, but the ribosomes are there. They're going along that mRNA transcript at the same time that transcript is actually being made. And then you have multiple ribosomes going along that mRNA transcript, making that same long section that, remember, has multiple codes for proteins, multiple sections of RNA for proteins. And then you can see that they kind of zoom in here to our RNA polymerase. Here's our RNA polymerase that's making the mRNA. And then here we have our ribosomes that are going to be making the proteins. So if you look over here, here is our DNA. I don't know why it says that here. So this is our DNA strand here, and then you can see our RNA polymerase is going along, and it's making many of these mRNAs here. And then we have our ribosomes going along, and they're making our polypeptides here. So, and you can see how many are doing this all at the same time. They're making them over and over and over and over again, because remember that our prokaryotic organisms are going to have a very small generation time. So it happens very quickly. Up here we have our eukaryotic organism, which we're more familiar with. Here's our nucleus, and in the nucleus we have our DNA. Then we have our pre-mRNA, then we go through the 5' cap here, poly A tail. We go through some splicing. Then that's going to leave the nucleus, and then we have translation occurring out here with our different ribosomes, and then we have making of our protein. So that ramps up transcription, kind of a short one here, and we'll talk more about mutations and things in the next section.